Hello and welcome to this tutorial on sequence similarity search using BLAST. In this video, we will explore how to use the BLAST tool to search for similar protein sequences, understand sequence alignments and interpret BLAST results. We will also explore how changing the database and scoring matrices affects the results. Additionally, we will analyze homologous, orthologous and paralogous relationships. First, let's start with a brief introduction to BLAST. BLAST, which stands for Basic Local Alignment Search Tool, is a powerful and widely used bioinformatics tool. It helps scientists find regions of local similarity between sequences. BLAST compares nucleotide or protein sequences to sequence databases and calculates the statistical significance of matches. BLAST can be used to infer functional and evolutionary relationships between sequences as well as help identify members of gene families. In this section, we will perform a sequence similarity search for human acetylcholinesterase using the UniProt ID P22303. Acetylcholinesterase is an enzyme that breaks down the neurotransmitter acetylcholine, playing a crucial role in terminating synaptic transmission. Understanding the sequence similarity of acetylcholinesterase can provide insights into its evolutionary relationships and functional conservation across different species. Let's begin by accessing the BLAST tool. Open the NCBI BLAST website. The official web address for reaching NCBI BLAST is https://blast.ncbi.nlm.nih.gov/blast.cgi. So we are in the home page of NCBI BLAST. NCBI is having different types of BLAST, including nucleotide BLAST, BLASTX, T-BLAST and protein BLAST. So for this session, we are going to do the protein BLAST. So select this protein BLAST. So this is the protein BLAST homepage. Now we have to input our sequence for the initial BLAST search. So we are going to retrieve the protein sequence for UniProt ID P22303. So for retrieving the sequence, we'll go to the UniProt. So in my previous session, I have shown how to retrieve the sequence from UniProt. The official website for reaching UniProt is www.uniprot.org. So once you are in UniProt website, you have to give the UniProt accession ID P22303 and click on this search. So here we have the basic information about the protein sequence of P22303. As I already mentioned, it is acetylcholinesterase and the organism is Homo sapien. So we have to copy the sequence in FASTA format for doing the BLAST. So we can go to this sequence and isoform section where we have the protein sequence. So we need the sequence in FASTA format. So click on this download and the sequence is now in its FASTA format. As I discussed in my previous video, it starts with greater than symbol, which is the syntax for FASTA file format. Copy this sequence. Copy the sequence and go to the BLAST page and paste it here. So once you pasted the sequence, next we have to set up the search parameter. The first important search parameter that we have to set is the database. So we have to mention against which database we have to perform this BLAST. By default, so it is non-redundant protein sequence database NR. But the protein BLAST is having different types of the important databases, including RefSeq database, SwissProt, which is manually curated protein sequence database. We have the protein data bank, we have patented protein sequences, and so on. We can choose any of this. By default, it is non-redundant database. So once you chosen the database, the next optional thing is we can choose the organism. So here you have the option for choosing organism. So if you want to limit your search to specific organism, you can mention the organism here. You have to mention the scientific name of the organism. So if you want to do the search only against human, you have to write here Homo sapien. I am not going to give anything now. So the next is we can also change some algorithm parameter. So that is here. So we are not going to change anything now, but there are options for changing. The important algorithm parameters includes max, target sequence, word size. Matrix is another important parameter we can change. So by default, the matrix is Blossom 62. We are not going to change it now. We can also change gap cost. We can also use the filters and masking option. So as I mentioned that we will run this search with the default parameter without making any change. So ensure that you have selected this checkbox so that the output of the BLAST will be outputted in the another tab. So click on this BLAST. So we have to wait for the BLAST results to be generated. Once the results are ready, we'll interpret the results. We got the BLAST result. This is a BLAST result page. So it is organized in different tabs like description, graphic summary, alignments, and taxonomy. We'll explore one by one. So we are now in the default description output page of BLAST. So first we will review the list of sequences that have significant alignment with the query sequence. So as you know that our query sequence is human acetylcholinesterase. So these are the database sequences which are having some similarity with our query sequence. So each entry in this list provides a summary of the alignment, including the sequence ID, description, and alignment score. So we'll examine the alignment score, e-values, and identities. The alignment score here, which represents the quality of the alignment between the query sequence and the database sequence. Say for example, so the alignment score for the first hit is 1255, for the second hit is 1254, third hit is 1253, and so on. The e-value, it is also called as expect value, which indicates the number of alignments with a given score that we can expect to see by chance in a database of a particular size. Lower the e-value indicates more significant. So it should be closer to 0 or 0. So here you can see all the top hits, they're having 0. So which means that they are highly significant. So the next important parameter is percentage identity. The percentage identity shows the proportion of identical matches between the query sequence and the database sequence. So all the top hits, they're having 100% identity, which means that at sequence level, they are same. So from here you can see there is a drop in percentage identity, 99.67, 99.67. And if you scroll down, the identity goes down. Say for example, for certain sequences, we have 93% and so on. So we'll now try to analyze the graphical summary. So this is the graphical summary. So here it's a query. It's the length of the query sequence, and each of this represents the database hit. That's a similar sequence from database. So the graphical summary provides a visual representation of where the query sequence aligns with the database sequence. So almost all the database sequences are completely covered, which is completely covering the query sequence in this example. So here you can see some sequences which are not completely covering. So this region of the sequences are not covered with the query sequence. So this region of the query sequence is not covered by this database sequences. The color bar represents different level of sequence similarity. So for this example, we have red color for all the database sequences, but not necessarily you'll get the same result. If you change the query sequence, you may get different color here. The color representation is the intensity of the color indicates the alignment score. So if it is a red color, the alignment score is greater than 200. If it is a pink color, the alignment score is between 80 to 200. If you get any green color region, then the alignment score is 50 to 80. If you get blue color region, the alignment score in the range of 40 to 50. If you get black, the alignment score is in the range of less than 40. So luckily for this example, the alignment score is good for all the database sequences. So next we will explore the alignment for query sequence and each sequences of database. So here you can see, so here this is the query sequence and this is the first hit from the BLAST result. So if you look at the result, the score for this alignment is 1255 and the expect value E value is 0 and the identity is 614 by 614, which means that there are totally 614 amino acids which are aligned. All the 614 amino acids between the query and the subject, they are same. So the identity is 100%. So 0% gap, as you know that while doing alignment, there are chances to insert gap. But for this first alignment, the gap is 0. So like that, if you scroll down, so here you can see the accession number of the second sequence, the database sequence, which is aligned with query sequence. So for this sequence, the alignment score is 1254, the expect value is 0, and the identity is 614 by 614, 100% identity. The gap is 0% gap. So like that, you can see the alignment between the query sequence and the various database sequences in this page. So now that we have our initial results, let us explore how changing the database and scoring matrices can affect the BLAST result. Different databases may contain varying sequences and different scoring matrices can influence the alignment score and E values, providing deeper insights into sequence similarity. Next, let's change the database and scoring matrices to see how it affects the results. First, we will change the database. So we will repeat the BLAST search steps, but we'll change the database to SwissProt from non-redundant database. And then we'll compare the results with those obtained from the NR database. So we will open a new BLAST page here. So again, go to the protein BLAST. Now copy paste the same sequence here. Now we will change the database from non-redundant to SwissProt. It's a manually curated protein database. So we are changing only the database here and run the BLAST again. So now we got the BLAST result after changing the database from NR to SwissProt. So you have to go through all the tabs for understanding and interpreting the results. So you have to compare the results with those obtained from the NR database. You have to observe and note any difference in the alignment score, e-value and identities. Now we will change the scoring matrix. So again we'll open another protein blast web page. So now go to the protein blast. So this time we will copy paste the human acetylcholinesterase sequence in its FASTA format here. Now this time we'll be using the non-redundant database and we'll make some changes in the algorithm parameter. So we'll try to change the amino acid substitution matrix from BLOSUM 62 to BLOSUM 80. BLOSUM 62 is the default amino acid substitution matrix in NCBI BLAST for protein. We'll change that BLOSUM 62 to BLOSUM 80. So that's the only change we are doing here, and do the BLAST search again. So it may take a couple of minutes for getting the output. So we got the results. So you have to go through the result page carefully one by one. So what we have to do is that you have to compare the results with those obtained from the NR database by using BLOSUM 62 matrix, because in this search we changed the BLOSUM 62 to BLOSUM 80. So you have to compare the results. So after changing the database and scoring matrices, make sure to answer the given questions to reinforce your understanding. In this section, we will retrieve and analyze the Miraculin protein sequence. Miraculin is a taste modifying protein found in the miracle fruit, which can make sour food taste sweet. Understanding its sequence and structure can provide insights into the unique properties and potential applications. So next, let's retrieve and analyze the Miraculin protein sequence. So go to the UniProt website. So now I hope you remember the web address of UniProt, www.uniprot.org. Just type the protein name, which is Miraculin. So we'll click this P13087, which is Miraculin. So just go through the basic information. The protein name is Miraculin. Organism, here they have given some fruit, miracle fruit. The total number of amino acid in this is 220. And if you look at the function of this protein, so here under this PTM processing, if you look at this, so there is a signal peptide in this protein. The amino acid from 1 to 29 is a signal peptide. Okay, so total number of amino acid of this protein Miraculin is 220, out of which 1 to 29 is a signal peptide. So the original amino acid starts from, the functional part starts from 30th amino acid to 220th amino acid. So we'll go to the sequence section and we will click on this download option. We will copy this sequence and we will go to again the NCBI BLAST. So I'm opening a new NCBI BLAST page. So in this, I'm going to copy paste this Miraculin sequence. So go to protein blast and click on this Miraculin sequence. So I have pasted the Miraculin sequence. So I'm going to use the non-redundant protein sequence database default, and I'm not going to make any change in the databases. The only thing is, while doing the BLAST, we do not need the first 29 amino acid because that is a signal peptide. So we'll be choosing the amino acid from 30 to 220 for our BLAST search. So here I am giving the query sub range from 30 to 220. So just click on this BLAST button. We got the BLAST result. So here you should go through the various tabs for understanding the results and interpreting the results as I discussed earlier. So now let's analyze homologous, orthologous and paralogous relationship from the output of this page. So homologous sequence or the sequences which share a common ancestry. So in this BLAST output page, we should look for high sequence similarity in the BLAST result. Say for example, if you take the first sequence, it's having 100% identity. If you take the second sequence, it's having 64.36. If you take the fourth one, it is having 64.17 and so on. If this sequence identity is more, then we can claim that they are homologous sequence. They share a common ancestry. That's how to find out the homologous sequence. Next, coming to the paralogous sequence. Paralogous sequence or homologous sequence with the same species that arose from gene duplication events. So we have to look for sequences within the same species with high similarity but may have diverged to perform different function. So in this, we have to look for the proteins from the database which are having higher percentage identity, sequence identity, but they are from different organism. We can claim those as paralogous sequence. So coming to orthologous, orthologous sequences are all also homologous sequence, but they are present in different species. They evolved from a common ancestral gene. So we have to look for sequences from different species with high similarity and similar function. So this is how we can find out whether the database sequence is a homologous sequence or paralogous sequence or orthologous sequence. So once if you complete this, make sure to answer the given questions after completing these steps to reinforce your understanding. Now let's move on to some self-evaluation task. They have given 3 tasks. The first task is retrieve the human and guanidinic insulin sequences. The first question is you have to retrieve the sequence of the human insulin gene and the guanidinic insulin gene in FASTA format from the NCBI website. I hope you can easily do it. The second question is what is the sequence length of the human insulin gene, and the third question is what is the sequence length of the guanidinic insulin gene. The task 2 is retrieve chimpanzee cytochrome C sequence. The first question is retrieve the nucleotide sequence in FASTA format for chimpanzee cytochrome C using the taxonomy browser of NCBI. What is the accession ID? What is the gene ID? What is the protein ID? What is the nucleotide sequence length? What is the protein sequence length? The third task is find beta lactoglobulin sequences. The first question is find 4 beta lactoglobulin amino acid sequence from different species similar to bovine. Scientific term is Bos taurus in FASTA format and GenPep format from NCBI. What is the e-value for each similar sequence? What is the sequence identity or similarity for each pairwise alignment? Which organism is found to be most closely related structural homologue to Bos taurus concerning beta lactoglobulin? So I hope that all the questions are simple and you can try it on your own. And by completing this exercise, you gain hands-on experience in using BLAST to perform protein sequence searches. You learn how to set up BLAST searches, interpret the results, and understand the significance of sequence alignments. Additionally, you explored the impact of changing databases and scoring matrices on the results, and learn to retrieve and analyze protein sequences from UniProt. The self-evaluation tasks further solidified your understanding of sequence retrieval and comparison using NCBI resources. This knowledge was essential for conducting bioinformatics research and analyzing protein sequences in various biological contexts. Thank you for watching. Don't forget to like, subscribe, and leave a comment if you have any questions. Stay tuned for more tutorials.